Awesome. All right. Well, we're live. So thank you to all of our participants who signed in today for putting up with a few technical issues that we've had getting started, but I think we're good to go now. I will not bite into too much of the time of our space talk today. I'll just introduce myself and our organization. So I'm Dave Fisher. I lead the University of Chicago Space Network, which is a university-associated alumni group. Our goal is to connect alumni and current students who care about space and the space industry to understand how to work together, how to network in the space industry, how to share our knowledge, and especially how to provide business opportunities, networking opportunities, and growth opportunities for everybody involved in this. So I affectionately call it Maroons in Space, and really happy to have you all here today. I'll say that in future, maybe we won't time this while an exciting Bears game is still going on. But let's go ahead and dive in. I'd like to introduce my fellow board member, Ian McCann, who is our Vice President of Programming, who has organized the Space Talks to do the introduction to the Space Talk concept and to today's speaker. Ian, over to you.
Thank you, Dave. As Dave has said, I am Ian McCann. I am the board's VP for programming. This is our second Space Talk. Last month, we had the honor of hosting Holly Heifel and Kishan Raghunath from Slingshot Aerospace to talk about their business with the Space Network. And this iteration of the Space Network features a wonderful company that I have had the pleasure of being a part of called Offworld Biotech. What we do is we genetically engineer microbes to stimulate biomanufacturing of materials, especially insulin and other drugs in microgravity. And I will introduce the CEO of Offworld Biotech, Harley Jackson, shortly. I will just say that Harley and I met at this wonderful institution called Mars University, and we and several other co-founders bonded over a shared interest of Mars settlement and exploration, as reflected by Harley's shirt. And we decided to get together and start Offworld Biotech quite a few months ago. And I'll let Harley take over from here and talk more about that. Thank you. Over to you, Harley.
All right. Yeah. Thank you, Ian and Dave. And really appreciate the opportunity to talk here. The University of Chicago's Alumni Network. Yeah. So like Ian said, Ian's a co-founder with me, and we have 6 other co-founders. Yeah, I guess I should go ahead and go to the slides, but yeah, basically, what I want to talk about is I first want to give an overview of the company in general. And then if we have time, I can briefly cover some results that we presented at the Mars Society Convention, a kind of pilot study that we did using E. coli. So yeah, let me see if go ahead and share my screen. Okay, yeah, so Offworld Biotech. So just a little bit about the company here. So yeah, we have the company is founded by people with a kind of unique overlap of expertise in space technology and biotech. And we have a shared interest and belief that biotech it's like one of the crucial pieces of infrastructure for deep space missions and space settlement. We're a distributed team right now. I'm in California, Ian's in Chicago. Our lab is actually at Western Michigan University School of Medicine. And that is where our chief scientist operates, Dr. James Seacrest. So he has a full wet lab. He can run protocols. He has like 40 years of experience in the space, and he has, he's run FDA trials, things like this. And he actually has the RPM device, which is applying a selective pressure to our strains of bacteria. And I'll actually show that in a video on a later slide. And yeah, the main mission we have right now is we're engineering strains of microbes that are suited to microgravity and other non-Earth gravity environments. Right now, we're focused on microgravity because lower Earth orbit is, it's the closest, it's the closest thing, and it's the one that is the nearest on the horizon time-wise. There are going to be commercial space stations available, as well as there are companies doing CubeSat experiments, which will be fully contained. So that's where we're focused right now. The machine we have also can be programmed for lunar gravity already. I mean, there's a pre-configured setting, and the equations have been worked out for Mars. The code has not quite yet. That's still in development. But if we were to have any clients who have an urgent need for Mars gravity, let us know. We'll go ahead and get that done for you. Yeah, so why space biotechnology? So, on Earth, a lot of us, probably take for granted that we actually live in this awesome biosphere, closed loop system that kind of takes care of things like recycling of water for us and waste management. I mean, we have to augment it for cities and things because of population density, but, the Earth kind of already does a lot of this work for us. But when you go into space, whether that's in a spacecraft, or if you're trying to build a space settlement, we are going to have to make the closed loop system ourselves. So that's what we mean by the biological infrastructure. Bacteria and autotrophs can do a lot of work for us in space, like some of them are things they already do on Earth, like human insulin production, recombinant protein production, human growth hormone. There's a lot of other pharmaceuticals that are actually made with genetically modified bacteria. And things like algae, for example, could be a food source, or spirulina. There's also waste treatment. There's also converting CO2 to oxygen. Yeah, and the key idea here is biology behaves very differently in space than it does on Earth. And probably, I don't want to say it's the biggest one, but for certain reasons, one of the most important factors is the gravity, how that affects what biology does. Yeah, so here's some considerations of environmental conditions that will affect the way biology behaves, the way it develops, the way it reproduces, especially at the cellular level. So you have gravity, magnetic fields, radioactivity, air, temperature. The thing is, the reason why gravity ends up being really important is, as right now, we don't have a good way to control it. We have theoretical ways, like an O'Neill cylinder that rotates, or maybe like sort of simulating gravity with something like a magnetic boots, but this still wouldn't address the problem of the actual cells experiencing gravity. So in the near term, what we need is biology that behaves in the gravitational environment as it exists, but it still does the job that we want it to do, whether that's making pharmaceuticals or otherwise. Yeah, so basically, one thing that's very prevalent in the research, and this is related to the study we recently did, is there are major microbial phenotypic changes that happen in microgravity. So some like immediate problems that brought attention to this was astronaut health, for one. So astronauts would get yeast infections and other bacterial infections in space, and when they took antibiotics that should control it, it did not. And basically they were looking at having to dose up to a level that would probably be toxic to the astronaut, or just kind of wait it out and until you get back to Earth. So this led to people trying to understand, why is this happening. So there was one really good study from that was done in partnership with University of Colorado, which we cited, where they actually had like a case. They had case samples of E. coli on the ISS, the International Space Station, and then they had controls on Earth in Earth gravity, and they could measure all these differences like cell wall thickness and diameter of the cells, clumping, and then also after they clump, they would secrete a lot of biofilm, and then they could see these phenotypic differences. They're statistically significant. They're actually markedly different. The growth rates go way up for E. coli. We've also seen this in our lab when we simulate microgravity. And the other thing they noticed was if you infused antibiotics into the culture in space, it took a much higher dose to kill the bacteria than the control sample on the surface of the Earth. So, okay, and another one was materials degradation. So a lot of the bacteria that's getting kind of spread in the ISS, it's very difficult to clean it up once it's kind of scattered around on surfaces. And because of the bacteria being like somewhat almost becoming something like super bacteria in microgravity, it also degrades a lot of materials where it wouldn't, these materials wouldn't get degraded on the surface. And this is pretty much just from, corrosion and from the bacteria. Yeah, so gravity, basically, gravity shapes life at all levels. Whether you're talking about molecular and cellular, tissue and organs, and at the holistic organism level. Like one thing that was pointed out in one of the courses we took at Mars University was when you go to space, you'll actually have blood like rushing up to your like heart and head, because we're evolved to be standing on our feet with a gravitational pull down. So this eventually goes away, but when you first get there, you can actually have like swelling of the brain, swelling of the heart, and it makes things really uncomfortable. If you aren't already uncomfortable from just being in microgravity. Yeah, so let me just say there's different ways to simulate microgravity on Earth. So one is neutral buoyancy. There's magnetic levitation. There are things like drop towers or parabolic flight. So if you've ever heard of zero G, these are the companies where you might see videos of people like floating in the middle of an airplane. And I think usually that's the company zero G. What they're doing is they're using the parabolic effect to basically create microgravity in an isolated area for a short period of time. I think like 90 seconds is about the max that they can do. And then you have these things called clinostats or random positioning machines. So they're a little bit different. But what these things are doing is they are rotating something in a manner that induces a continuous parabolic effect that can last for very long periods. So for example, basically a clinostat will do this in 2 dimensions. They tend to look like a cylinder spinning, and then there's another spin happening on the edge. RPMs, or random positioning machines, some people call them 3D clinostats, but really a RPM is it's moving in 3 dimensions, and the idea is that you can have this microgravity effect without like what's the right word? Like the centrifugal force that happens with clinostats, because clinostats are continuously spinning around one axis all the time. But with the RPMs, they're not. They're random or pseudo-random. And yeah, so biogravity is a company of our partner, one of our co-founders, which is Dr. James Seacrest. And he has an industrial grade RPM that is the one we're using to develop. So let me go ahead and just play a little video for you guys, so you can see what this thing looks like. So this is, inside here, you can see it's kind of tied in. There is a T25 flask, and inside that flask, you can have a sample of cells in a medium. In our case, it's E. coli right now. He's done work with human cells as well. But basically this thing can run continuously for, a long time, weeks. our studies, we've been running it for about 24 hours at a time. And it's inside of an incubator. So that's how that looks. Let me see if I can't get out of this video. All right. So yeah, this is what we're working on. And then there's, the real deal. So these are simulated microgravity. The other way you can access microgravity is really send your stuff to space. Some examples are International Space Station or commercial space stations, which really are being made. There are companies such as Axiom and a few others. I believe another one's called Orbital Reef. So yeah, this is really going to be happening very soon. And another one that I actually just discovered a couple of days ago, and I actually didn't add it to the slides. Another one is there are companies that are starting to make CubeSats where you can have a self-contained experiment inside of a CubeSat, which will execute automatically once it's released into space. And this is really interesting because it's going to be probably more affordable than sending something to the ISS. When you send something to the ISS, you have to pay for astronaut time, which is extremely expensive. I believe it's like $130,000 an hour. So literally, if you need someone to do something for 5 minutes, it still costs a lot of money. And the other reason is when you want to go to the ISS, there are specific times scheduled, and you're probably not going to get other times. So, all right, can you guys see? I don't know if you guys can see this update, but sorry. So I have this blurb in the middle of my.
We can't see it if you got.
You can't see
an
it?
error box on yours. We're good. We don't see anything like that.
Okay, great. I don't know how to get rid of it. Okay, so I'll just pretend it's not there. Yeah, so the CubeSats, yeah, the other thing is you won't have to schedule, you won't have to build your experiment around a scheduled ISS visit. You just need a launch, which that's still a constraint, but it's less of a constraint. Yeah, so let me continue from here. Yeah, this thing.
Hey, Harley, I know sometimes well, that did work. You went to the next one.
Yeah.
If you're more comfortable with it, it's okay if you'd like to just stop sharing your screen for a second, deal with the dialog box, and then share your screen again. I think we could roll with that.
Okay, I think I got it. So I was able to get it out of the way. Thank you. Yeah, so basically our solution is we offer the Explorer X biogravity platform, which was basically, it was built in Australia with a team called Explorer, and biogravity is the US arm. And yeah, it's the industrial grade RPM. It has a software kit, a GUI that you can use, or a GUI, where you can basically input different parameters and then run your RPM, and and then they provide pre-configurations that match, for example, microgravity or lunar gravity. Those are the two that are available right now that are probably the most useful. But you could actually, like, if you wanted to work out Mars gravity, you can do it manually. It's very configurable as it exists. So it can be programmed for different gravitational unloading potentials. And the other idea is, like, why would you use an RPM? Well, sending things to space it's expensive. It takes a lot of time and coordination, and it slows down your iteration time. So in the case of Offworld Biotech, what we're doing is we're taking microbes, we're subjecting different candidate strains to a selective pressure, and in this case, that's microgravity. And then we're measuring how they behave. And then what we're doing is we're taking another step where we try to identify why do these two strains behave differently, and how can we modify them to make them behave more like what we want it to do. So if in our case, if we wanted to make human insulin, we want to make it produce even more human insulin in microgravity. And then we, after we generate more candidates using some genetic modification, we then repeat the cycle. We subject them again to the RPM. So that iteration cycle is much shorter if you're doing it in a laboratory with simulated microgravity, as opposed to if you were to send them to space every single time. So what we're looking at is use simulated microgravity for prototyping and development, and then use space for validation to make sure that the different candidates you've come up with in fact behave in space the way that you want them to. And why did we pick E. coli as the main microbe we're working with right now? Well, E. coli is very favorable for producing recombinant proteins, other human biologics. It's easier to culture and evolve as compared to other microbes. The requirements for media are pretty lax, and they grow pretty quickly. They culture pretty quickly. So we're only subjecting ours to microgravity for 24 hours and getting a lot of results in that amount of time. And yeah, and so we're working with several strains right now. Some have been modified to express certain proteins. Like one is expressing short chain variable fragments, and that's used in vaccines. And that's in partnership with North Carolina State University. They shared that strain with us with the MOU, and we're testing how that behaves in space, for example. Yeah, and basically, so the products we're offering as a company. So our longer term is we're going to basically have a library of patented microbes that we can license to biomanufacturing companies in space. These companies are already in existence. They're just they're getting to the state where they're actually going to produce things on commercial space stations. And the idea is that we will be able to license, we will already have the microbes ready that they need to, for example, make human insulin or other products, and they just license it from us. That's a longer term revenue model. The short term, one major candidate for short-term revenue is to offer end-to-end microgravity research. So right now we can already offer the simulation component because of Dr. Seacrest's lab, but we also have partners who, in fact, tomorrow I'm talking to the one partner who does the self-contained CubeSat experiments, and then another partner does payload brokering to the ISS. And the idea is that along with our partners, we can basically offer a service where, say you want to do some kind of research with microgravity, whether you're, I don't know, PhD student or a professor or just an independent researcher, or even a company. What we can do is, because of our expertise and our access to partners and equipment, we can help you devise a experiment that is sound and execute the experiment, including sending your cellular biology to space. Usually something small, whether that's bacteria or it could be skin cells, or whatever kind of experiment you might come up with. Yeah, so this is what we're offering, and yeah, basically. So I'm not sure if it's relevant to go through the rest of this stuff. But yeah, I think this is pretty good. I don't know, Dave, do you think we have time to look at what happened with the E. coli briefly?
What time is it? Oh, I think so. Let me just, before you dive into that, we've had one question come into the Q&A. So let me actually ask you the question that came in. It came from Jonathan Handler, and it says, how long are the transients for E. coli, hearts and brains? Order of magnitude, is it fractions of a second, seconds, minutes, hours, days?
Okay, how long are the transients for E. coli, hearts and brains, order of magnitude?
I think for the body, the human body as a whole, I think the response, the adaptation to microgravity peaks at around a couple within a month or so for long-term human spaceflight. Of course, obviously stuff like bones never peaks off, so you'll have to account for that. But that's just the human body. I don't know about bacteria in particular.
Let me just remind the audience as well, please use the Q&A tool, throw questions up into here, and I'm going to act as sort of a moderator and ask Harley these questions. But Harley, go ahead and go on to the next part of your presentation as we collect up more questions.
Okay. All right. So yeah, this is actually a slide. I'm not going to go through the whole slideshow, but this is what we presented at Mars Society Convention recently. So yeah, the study was done. This is our team here. Dr. James Seacrest, me, Margarita Balali, Ian, Terry Trevino, Ben Lyons, and Andrew Lord. And biogravity is they own the actual RPM technology, and then Explore develops that. I mean, biogravity, yeah, basically can work with like it. Yeah, they can work with the RPM, and they can consult on experiments that involve it. And yeah, and some of the basically the clinical part, especially like the study design was done by some postdocs at University of Technology Sydney. And yeah, so I'm going to go through some of this kind of, I'm going to kind of skip a lot of this because what I want to do is I just want to show you the actual results and some of the motivation. So yeah, so here's some of the things that we saw in the literature. Basically the changes in cell morphology, which I think I did mention earlier. But yeah, you get smaller cell size, thicker envelope, extracellular vesicle formation, development of an irregular cell shape, so they kind of elongate, and then they cluster more. They secrete biofilm production. They secrete biofilm, and then all of these in aggregate seem to lead to like an increased antibiotic resistance. I don't know that it's totally like known for sure that there's a cause effect relationship between these two things, but it does like appear to it appears to be a reason why. So yeah, this is from another study, but that not one that we did, but one that we kind of benchmarked for our pilot study. And this is the name of the paper. And yeah, you can see how like gentamicin being infused would basically on the Earth sample, you would still have this high amount growing, and then it would just totally fall off at 100. But the space sample still persists, and it does start declining, but it takes much more concentration. So that's the antibiotic resistance portion. One thing I will say is that when we did our study, we weren't able to replicate that part. Like we still saw the crazy growth curves and all that, but the antibiotic resistance wasn't replicated. So the question becomes whether their study had a flaw or whether ours did. And this is the reason why we actually want to, we plan to replicate the study we did in the lab in space to find that, to try to check that question. But yeah, so you see some differences in cell volume and then cell length and cell diameter. So this is related to, this graph is looking at the shape, the elongation, and yeah, the cells in space, they tend to be much smaller, but more of them grow and with thicker cell walls. And then here are some pictures. So this is where you can kind of see the clumping going on. And then these are the extracellular vesicles that form. Yeah, so what, how did we devise our study? What we wanted to do is we wanted to compare the growth in, similarly to what they did on the ISS, but our case samples are in a simulated microgravity, namely the Explore X biogravity platform. And yeah, so this is talking about some things I think I already covered, so I'll go ahead and skip this. but yeah, well, I guess I should say, yeah, the biogravity platform was set for microgravity at 10 to the -3 gravitational unloading. So this is as compared to Earth gravity. I think this is the same video I showed, so sorry about that. So here's the controls and the cultures that we had. Basically, we had we used TSB media, and then we had this Zuno antibiotic spray, and then so we had one pair that doesn't have it, and then one pair that does have the spray, and then we did it again with the gentamicin, which is what they used in the study we were benchmarking. And this was, we just did, you know, one dose, or yeah, one dose, basically the 1 milligram. We didn't have like several different concentrations. So in the, yeah, we used T25 flask. That was what you saw strapped inside of the RPM. And yeah, we ran ours for 24 hours, and we measured the cell growth, OD 600 nm. And we just did a basic ANOVA analysis of comparing two different populations. Yeah, so the results were, we definitely saw, like, we took measurements over time, and you can see that the growth of the E. coli and microgravity outpaced pretty significantly. And actually, regarding the question I got earlier from Jonathan, I think it just clicked what you're asking. Like, I think what Jonathan's asking is, if something, you know, if you go into space with, say, these cells and they behave differently, is that a continuous effect, or is that just some kind of like effect that happens at first, and then once they adjust, they go back to a behavior that's comparable to being on Earth? That's actually, assuming that that's the question you're asking, I think that's a great question, at least at the microbiome level, because it'd be important to think about for us, because we're engineering microbes. As far as like what happens with humans and stuff, yeah, I would have to check the literature on that. All right, so we did some measurements where we're basically looking at like the, you know, these are the antibacterial coated samples, and then here are the non-antibacterial coated. So this is what I was saying about the not reproducing the variable behavior with antibiotics. So like in space, they grow away, or not in space, but in microgravity, they grow away faster, but the antibiotics was the coating anyway, the spray was still killing them off. And actually, so was the antibiotic. Yeah, so this is something to think about, is like whether there's a flaw in our study, or maybe it's because of concentration variables. And then you can see some differences in fluorescence. It's a lot higher in the microgravity, and how much absorption goes on. So yeah, basically, definitely there's some, you know, the microgravity device is replicating some of the behaviors, not all, meaning possibly not the antibiotic resistance. But yeah, we want to validate that by sending it to space. And here's some photographs of the samples. So this is gravity without antibacterial coating, gravity with the coating. This is the spray. And then these two are the gentamicin. Yeah, and these are, sorry, what was this last one? Oh, sorry. Okay, these are gravity. Sorry, this is all just a spray. Gravity with spray, microgravity with and without spray. And then these are the ones that are gentamicin. And yeah. So I think I kind of mentioned it, but yeah, the cultures grew substantially faster in simulated microgravity. So we think that this could also have possibly an industrial application right here on Earth. If, you know, if you're wanting to manufacture something with E. coli, and we can use simulated microgravity to accelerate the speed. This could possibly speed up an industrial process. Right now, the machines are only big enough to be used for lab experiments, but they could probably be scaled up. So this is something we've been thinking about. And yeah, the other thing is we think that some biomanufacturing processes that are accelerated and simulated microgravity, those could be good candidates for being moved into space earlier on. So, there's a push right now to move heavy industry into space because, there's there will be less of a load on our biosphere as far as like pollution goes and things if you're making these things in space. And ones that perform better in microgravity, those would be like candidates to be the first ones to go up, probably. So these are a couple takeaways we had from our pilot study. And yeah, for future research, yeah, the main thing we want to do is we have some other strains that we've been working with in the lab, and we have data on that. We didn't present them at this study or this conference yet. We have another one next month in Houston. I'll probably give a talk about one of those there. But the main thing we're looking at right now is we want to validate in space. So whether that's on the ISS or as a proof of concept in one of these like self-contained CubeSats. And I think, yeah, this is our contact information. Here's like a little bit more information on our team. So yeah, we're all on LinkedIn, Offworld Biotech, and yeah, got a really awesome team here. Dr. James Seacrest, his background is pharmaceuticals, FDA trials, and he also knows, he also has a lot of background in like space research, very knowledgeable. My background's professionally, I've worked in big data software. I still work for LinkedIn and doing like a lot of root cause analysis, things like that. But yeah, I have a lot of education in space and really interested and passionate about this. And then we have Margarita, who is in Europe, and she is probably going to head up our arm in Europe because there's a lot of, the ESA has a lot of funding for these kind of projects, but your firm needs to be based in Europe. So basically she can handle that. She's in Greece. And then we have Terry Trevino, who's like a serial space entrepreneur. He has another startup that does electromagnetism. And he does like algae experiments. You'll see him if you go to any space conference, you'll probably know who Terry Trevino is. And then yeah, Ian, who's on the call here, excellent at microbiology and also just generally anything regarding space. And Ben, so Ben's background is evolving bacteria. So he's very helpful with the actual process that we're working on, and he specializes in getting E. coli to grow faster, to mutate faster, and all that, which is all factors. And he also has a really good network in that field because he's been doing it for about 10 years. And then we have Andrew Lord, who's our finance guy. And the thing is, the rest of us are kind of like mad scientists, mad engineers like this. And then Andrew's the guy that yells at us and says, hey, but how are you going to make money with this? Which is a great question because we're doing a business. So that's a question we got to answer. And yeah, so, and he also has a lot of background with space related policy and risk management. So. All right. And yeah, I think that's it for the presentation.
Harley, thank you so much. This was great. I've been writing down some questions I've got, and I think Ian may have some contribution he'd like to make too. So Ian, please come and rejoin us here. Harley, the question I have, you asked at the end, it's a variation of how are you going to make money at this? I wonder if you could share a little bit about your fundraising journey, where you are in fundraising, and also if you could talk a little bit about customer discovery, to understanding a lot of that stuff's proprietary with new startup companies, but anything that you could share, I think, would be really valuable to the group here.
Yeah, so I think the biggest challenge that a company like ours faces is that the core payoff is, you know, anywhere from 5 to 10 years down the line, because we're talking about a pure licensing model where you have to have like you know, a patented library of novel organisms which you can license out, but all the, you know, R&D and supplies and everything that leads up to having that library all cost money. So the question is, how do you get funding before then? So these are the kind of companies that, like, you might call them like moonshots. Google funds a lot of these kind of companies where they know that they're not going to get any return in the near future, but they just fund it anyway because they think it's important. But we've been looking at short-term revenue models. So one was like what I mentioned earlier, where we actually offer microgravity research services, where, you know, like someone that's procured a grant, or for example, for some kind of research that's going to support NASA or other space endeavors, but they don't have all the equipment, you know, they don't have the RPM, or they don't have the network. We can kind of broker, we can basically take a lot of the work off their plate and get all that part done, where they focus on the actual, like, say, the paper itself, the study design, and getting that published. So yeah, that's one thing. There's products we can probably make, but yeah, for their fundraising journey, I think what I've noticed is, like, there's a lot of interest amongst investors that I've talked to, but I think this is the kind of thing that you need a very specialized investor who kind of understands what we're doing, understands biotech. I have been connected with some of these individuals, and then, you know, they're going to want to, even after you present, you know, your pitch deck to them and everything, each one of them is going to want to know different, they're going to want something different than the other investors want. So almost every time you're going to have to go fetch, you know, some other requirement that they give you, and then get that ready and then go back. You know, so there's a lot of back and forth. It's a lot about, like, even if someone isn't going to be a direct investor, sometimes you can find some other value that you can bring this person, and then what they can do is they can refer you to the right person, or, you know, yeah. So it's like, it's a lot of networking. So I'd say, like, it's super important that you take opportunities to meet people that would otherwise be difficult to meet, is, you know, like busy VCs and people that are in good positions to help you out, point you in the right direction, and, you know, even give you advice. So I don't know if that's, like, totally answering the question, but hopefully.
Oh, yeah, thank you very much. I realized I don't actually have to read out loud the Q&A questions that are put in, since I think you can see them too. Would you like to address UNPA's? I apologize if I said that wrong. Question, Harley?
Yeah, so I'm looking at that. I'm interested in the way nanotechnology and transmitter receivers work in microgravity. Is there a big difference? Is there a way to ensure security and minimize interference on communication between Earth and space? Yeah, this is so I think this is a little bit outside of my expertise in microgravity. I've been focused on biology, actually really focused in on, like, the kind of work we're doing. But I think that's, like, a really good question, an important question. And you know, it's even possible that Dr. Seacrest might be able to answer that, or if he can't answer that, he knows somebody. So if you'd like, shoot us an email, info@offworldbiotech.com, and we'll see if we can help you out.
I mean, just to jump in a bit, I don't know if like anything in I don't think light is especially affected by gravity, especially not Earth gravity. But just off the cuff, I think atmosphere might be more impactful on electromagnetic radiation and light and radio communication than anything specifically gravity with gravity specifically. But that's just me off the cuff. But thank you.
Right. All right. And I think the other questions are from Jonathan, which I believe I already addressed. So,
Yeah. Harley, I'm curious if you've looked at tagging up with any of the startup accelerators that are out there for space companies, if that fits into your business case at all.
yes. Yeah. So yeah, that's something we're considering because, so to give a little bit more background on myself, like I have done startups in the past, including recently I helped get a nonprofit off the ground called Getaway Growth. And we went through an accelerator, or yeah, we basically went through like a nonprofit accelerator. And yes, I think these are crucial tools because yeah, a lot of the info they give you, yes, you can go find it on your own, but the thing is they're putting it right in front of you, and it makes things happen at a much faster pace. And they also tend to have network, which can help you, which is crucial, like I was mentioning earlier about fundraising. So yes, what I've found so far is a lot of the ones that support like our kind of business space ones, they tend to have like they tend to not be available all the time. A lot of them start next year. So like, yeah, we're looking at that. And also if you know of any good incubators that we should look at that would be interested in this kind of like, almost literally moonshot type of business, let us know. Be happy to take a look.
Yeah. Yeah, absolutely. And Harley, why don't we connect up offline, and I can give you some more information. But for the benefit of everyone on this call, one of the people that we had as a panelist at a space financing discussion we had a few months ago is Ellen Chang, and Ellen runs an accelerator that I think would actually be a pretty good fit for Offworld. So just a little plug for my friend and colleague Ellen, who does a really awesome job with space startup with an accelerator. I don't see any other questions coming in, Harley, and we've certainly run through the ones that I've written down. So let me just ask again, anyone online, if you have any Q&A, this is your chance to do it as we begin to wrap up a little here. Harley and Ian, I'll turn it over to you for some last thoughts while we wait for any straggler questions to come in.
I'll just speak as a VP for programming. That I'd just like to thank everyone for coming here again. Between 15 and 17 attendees were here, similar to last month's total. And I am proud to see the Space Talk venture become a more sustainable and viable long-term programming opportunity for the Space Network. Thank you.
Awesome. Harley, any closing thoughts?
Yeah, right. No, I think yeah, I think I covered everything I had on my mind. But I really appreciate everybody that came for the talk and the opportunity to present here. Very grateful. Oh, it looks like maybe someone typed another question. Oh, okay. Question. We have the current issue of, this is from Teresa Daniel. We have the current issue of space debris. Thinking about your project, how do you think your company will mitigate this issue so that your experiment does not collide with objects? Yeah, so this is a great question. So like there's two possibilities for how we're going to validate. One is if we go to the ISS, and then that that would be returned in a payload. So I don't believe that route would lead to space debris. So for the scenario with a CubeSat, I really appreciate this question, because I'm gonna talk to them tomorrow about that. We have a call tomorrow, and we're gonna be discussing different like factors. Like one question I have is whether we're actually able to recover the CubeSat, or if this is something that just like burns off in space. This is something I don't know yet, so I'm gonna actually ask our partner about that. And then I will also bring up, how do they guarantee that this CubeSat, while it's orbiting Earth, isn't causing problems and colliding with other objects. But yeah, so this is great. I'll make sure to bring that up.
Super. Hey, well, let me just echo Ian's comments. Thank you to all the audience members who took part in today's conversation. Harley, thank you so much. A Sunday afternoon, coming on and talking to us about your company, your passion. This is really fascinating stuff. Wish you absolutely the best of luck going forward. And thank you once again. With that, with no additional questions coming in, we'll go ahead and close. Have a great rest of your Sunday, everybody.
All right. Thanks so much. Have a good one.
Thank you.